Hello and welcome back to Medical Terminology. This week we're going to discuss Chapter 10, the blood system. So let's get started. Learning outcomes. 10.1, we're going to identify the parts of the blood system and discuss the function of each part. 10.2, we will recall the major word parts used in building words that relate to the blood system. 10.3 will identify the common diagnostic tests, laboratory tests, and clinical procedures used in testing and treating disorders of the blood system. 10.4 we will define the major pathological conditions of the blood system. 10.5 we will explain the meaning of surgical terms relating to the blood system. 10.6 we will recognize common pharmacological agents used in treating disorders of the blood system. And finally, 10.7 we will identify common abbreviations associated with the blood system. All right, 10.1. Let's discuss structure and function of the blood system. So blood is a complex mixture of cells, water, and various biochemical agents such as proteins and sugars. We'll get a little bit more into that. Blood has many functions, including transportation of life-sustaining nutrients, oxygens, hormones to all parts of the body. Transportation of waste products from cells of the body to prevent toxic buildup. That's what our lymphatic system helps with. Helping to maintain the stability of fluid volume that exists within the body tissue. The blood system also is important in helping to regulate the body temperature. The blood volume of an average adult is 5 liters. This may vary depending on the body size. Blood comprises about 8% of total body weight, and blood loss through bleeding or blood donation takes 24 hours to replace. Blood circulation. Here you can see everything usually starts with the heart, and then oxygenated blood flows through the arteries, into the capillaries, and where the gases are exchanged. Then the deoxygenated blood goes through the veins back to the heart, which delivers it to the lungs for reoxygenation. Once it's reoxygenated, it goes back to the heart so it can get pumped out to the arteries and do the whole system route again. All right, let's look at some combining forms. So here we have agglutino, which means clumping. I like to think of the glue part in it. Think of that how that would clump stuff together, right? It'll hold it together. Then we have eosino. Eosino means red or rosy. Urethro means red. Hemo or hemato, that is our word part for blood. Then we have leuco, that's one of our colors, that's white. Phago means eating or devouring. All right, first let's discuss plasma. Plasma is the clear liquid portion of the blood. 55% of total volume of blood upon separation, which is made up of 92% water and 8% organic and inorganic chemicals such as protein, glucose, nutrients, gases, and electrolytes. Plasma proteins such as albumin, globulin, fibrogen, and prothrombin assist in the clotting of blood. Platelets. Platelets are also known as thrombocytes. You might hear of a thrombus or an embolus even. All these have to do with the platelets. So platelets are the cells that circulate in the blood that are needed for blood clotting. They're used for wound healing. It's produced in the bone marrow, and it's the first to react to an injury to the body. Structures and function terms. So we have albumin, globulin, fibrogen, prothrombin, gamma globulins, electrophoresis, plasmapheresis, coagulation, thromboplastin, fibrin clot, thrombin, and heparin. Here we'll talk a little bit about blood cells. So there are two types. We have red blood cells and white blood cells. Now RBCs is the abbreviation for red blood cell, and it carries oxygen. White blood cell fights infections, and you have multiple types of white blood cells. Erythrocytes are also your red blood cell. Leukocyte is a white blood cell. So you can see where we have our colors there. Urethro is our word part for red. Cyte is our word part for cells. That's our suffix. And then leuco is our combining form for white. Cytes, again, is our suffix for cells. And then we have thrombocytes, which are also known, you probably have heard them called platelets, but their technical term is thrombocytes. Then we have hematocrit and stem cells. Let's discuss a little bit of blood development. The first flow chart shows the process of erythropoiesis. During the process, initially a progenitor cell, the self-renewing stem cell, gives rise to the prourethroblast. The first or early urethroblast, remember blast means embryonic stage of development or young, so this is a young, immature red blood cell. Precursor is produced under the influence of the erythropoietin, or the EPO hormone. After the early urethroblast, later urethroblast, and then the normoblast is formed. In a further stage, the normoblast ejects its nucleus and gives rise to the formation of a reticulocyte, and then the mature erythrocyte, so that's a fully mature red blood cell. The second flow diagram shows the process of thrombopoiesis. During the process, initially a progenitor cell gives rise to the megakaryoblast. Further, the promegakaryocyte precursor is produced under the influence of the thrombopoietin hormone. Thrombopoietin also helps in the development of a megakaryocyte and mature platelets in the subsequent stages. The third flow diagram shows the process of leukopoiesis. This is the process of building those white blood cells. The process starts from the GM-CF progenitor cell, which further divides into myeloblast and MCSF monoblast. The myeloblast takes part in the formation of the GCSF promyelinocyte, which further develop in three directions from eosinophilic myelocytes, basophilic myelocytes, and neutrophilic myelocytes that ultimately develop into eosinophil, basophil, and neutrophils, respectively. MCSF monoblast further develops into MCSF promonocyte and ultimately the monocyte. For the development of lymphocytes, B lymphoblast develops into B lymphocytes and T lymphoblasts develop into T lymphocytes. You might know those as T helper cells. Next, let's talk about some blood types. The chart shows the following: erythrocytes, antigen A. Diagram shows antigens with round tips attached to the disc-shaped cell membrane. Antigen B diagram shows antigens with a conical tip attached to the disc-shaped cell membrane. Antigens A and B diagram shows antigens with both round tips and conical tips attached to the disc-shaped cell membrane. Neither antigen A nor B diagram shows a disc-shaped cell with no antigens attached. Now let's look at the plasma row. As you can see, type A has anti-B antibodies, type B has anti-A antibodies, and type AB, neither anti-A nor anti-B antibodies are present. And for type O, both anti-A and anti-B antibodies. The antibodies are illustrated as Y-shaped structures. So let's look over the different blood types row. Type A, erythrocytes with type A surface antigens and plasma with anti-B antibodies. Type B has erythrocytes with type B surface antigens and plasma with anti-A antibodies. Type AB has erythrocytes with both type A and type B surface antigens and plasma with neither anti-A nor anti-B antibodies. And finally, type O. Erythrocytes with neither type A nor type B surface antigens, but plasma with both anti-A and anti-B antibodies. Now let's talk about the Rh factor in pregnancy. Rh factor incompatibility occurs when an Rh negative woman and an Rh positive man conceive a child. The Rh negative pregnant woman has now exposed her Rh positive fetus. Cells from the Rh positive fetus enter the mother's bloodstream. Now the woman becomes sensitized, which leads to the formation of antibodies to fight Rh positive blood cells. It does not affect the first pregnancy as the number of antibodies is low, but in the next Rh positive pregnancy, maternal antibodies attack fetal blood cells. Learning outcome 10.2, word building in the blood system. Here you'll see we have eosinopenia, which is an abnormally low count of eosinophils. Then we have leukoblast. Remember, blast is that immature cell, so this is the immature white blood cell. Erythrocyte, this is a red blood cell. This is a fully formed red blood cell. Thrombocyte, a cell involved in clotting, so this is your fully formed platelet. Agglutinogens cause the production of agglutinin. Then we have phagocyte, which is a cell that consumes other substances such as bacteria. Hemodialysis, which is the external dialysis performed by separating solid substances and water from the blood. And lastly here we have a cytoplasm, which is the solution that fills the cell and that is enclosed by the cell membrane. 10.3 Diagnostic, Procedural, and Laboratory Terms. Here you will see that a CBC means complete blood count. We have blood chemistry, biochemistry panel, blood indices, and blood cultures. You'll see erythrosedimentation rate, which we'll also see as ESR in the charts. Sedimentation rate, which is just SR. Then we have white blood cell differential and red blood cell morphology. We have platelet count, PLT. Then we have partial thromboplastin time, which is the PTT. That's how long it takes for your blood to coagulate. You also see prothrombin time, which is known as PT, antiglobulin test, white blood cell count known as a WBC, red blood cell count as a RBC, hemoglobin, which is HGB, or you'll see it with a capital H lower GB, hematocrit, HCT, you can see it in both ways as well. All right, learning outcome 10.4, pathological terms. Many diseases and disorders have some effect on the blood, but they are really diseases of other body systems. For example, diabetes is a disorder of the endocrine system, but its diagnosis includes an analysis of blood glucose levels. Some other pathological terms you may see: dyscrasia, sickle cell anemia, polycythemia, von Willebrand disease. Learning outcome 10.5, surgical terms. Surgery is not generally performed on the blood system. There are, however, two exceptions: a bone marrow biopsy or a bone marrow transplant. Learning outcome 10.6, pharmacological terms. Medications may be used to treat blood disorders and diseases. Medications do not cure the disorder, but treats and prevents symptoms, such as anticoagulants, thrombolytics, coagulants, and hemostatics. Let's look at these terms real quick. So anticoagulant, we know coagulant is coagulation, right? So anti means that it's going to keep the substance from coagulating. It's going to keep it from forming. Then we have thrombolytic. Now, lytic is our word for destroying or destruction. So thrombolytic means it's destroying those thrombocytes or those platelets. Coagulants means it's the opposite from that anticoagulant. So in this case, we are trying to help it coagulate. Hemostatics, remember hemo is our word part for blood. Static means stopping or controlling. So we're trying to control the state in which the blood system is at. Learning outcome 10.7, abbreviations. So BASO is short for basophil. BMT, bone marrow transplant. CBC, we've covered this one before, complete blood cell count. EOS, eosinophil. ESR, erythrosedimentation rate. Remember, erythrocyte is a red blood cell. Notice it's missing a parenthesis here. And then PT is prothrombin time. And RH, rhesus. SR, sedimentation rate. Other abbreviations: HGB or HB for hemoglobin, mono for monocyte, NEUT and EUT for neutrophil, PTT for partial thromboplastin time, RBC for red blood cell count, and WBC for white blood cell count. Chapter review. 10.1. We discovered that blood transports life-sustaining nutrients, oxygen, and hormones to all parts of the body. It prevents toxic buildup, helps maintain the stability of the fluid volume that exists within the body tissues, and helps regulate body temperature. In 10.2, we discovered word building requires knowledge of the combining form and meaning. 10.3, diagnostic, procedural, and laboratory findings assist the healthcare provider in diagnosing medical conditions. Often used in combination, these tests lead to a final diagnosis and assist in the treatment planning. 10.4, many diseases and disorders have some effect on the blood, but they are really diseases of other body systems. 10.5. Surgery is not generally performed on the blood system, but there are two exceptions: bone marrow biopsy and a bone marrow transplant. 10.6. A variety of medications may be used to treat blood disorders and diseases. Medications generally do not cure the disorder, but rather they treat or prevent symptoms. And finally, 10.7. We learned abbreviations that are used to describe diseases, conditions, procedures, and treatments associated with the blood system. All right, everyone, thank you so much for sticking with me. That is all I have for you today. If you have any questions, please feel free to let me know.